cytoplasm and nucleus. And the first three constructs localize primarily to the cytoplasm. And the final, without the C-terminal tail, we can see, this is the zoom in of them. Without the C-terminal domain, we can see that the NBC1 construct does not localize properly to the plasma membrane. But once a full-length NBC1 is expressed, you get proper localization of NBC1. After this, when we decided to run some purification of the samples and send them to mass spectrometry, I'm not going to show you the results, but I can tell you that it was successful, the purification of the GFP constructs, and we decided to send 3 samples for mass spectrometry analysis, namely the GFP control, the N-terminal NBC1, and then full-length NBC1. Unfortunately, analysis of the constructs has yet to be carried out, but I hypothesize that due to proper expression purification, and MS analysis should return binding partner candidates. This is the conclusion for the first part. And now we go on to the second part, which is the phosphorylation site characterization. So, first of all, phosphorylations, they can alter a protein's activity and structure. So we hypothesize that NBC1 activity could be altered by these phosphorylations. And this could be elucidated by PHI measurements, which I'll go through later. Here we have NBC1 again, and several phosphorylation sites of NBC1 were identified by a study by Lundby et al., where they took 14 tissues from rats, snap frozen, and then through mass spectrometry analyzed peptides that were phosphorylated. Through this analysis, we chose 4 sites which were conserved across species. These are included here, namely it's serine, residue 89, 407, 411, and S111A. You see these 3 on the N-terminal tail, and this one's in the C-terminal domain. So how do we probe these phosphorylation sites? You can mutate into alanine, which removes the potential of phosphorylation on the site, whilst minimally altering the native structure of NBCN1. So these mutations, they were created before I started my master's degree, before I started my experiments, and then we created a HEC293 cell lines stably expressing wild type and these mutant NBCN1s. So was this successful? Here we have the heterologous expression of NBC1 in the HEC cells. On the top we have a representative Western blot, where first we have control, which are cells that have not been transfected with any extra NBC1, wild type, and then the 4 mutant variants of NBC1. A band is seen in the control, since there's an endogenous expression of NBC1 in these HEC cells, and we see an extra band visible in the rest of the transfected variants, which is the extra NBC1, which is a little bit heavier because it's the longest variant of NBC1. On the bottom we have the protein levels normalized to wild type. The only significant difference we see is control cells, which express, let's say, about 20 to 25% NBC1, which is the native NBC1, and then the other variants. There's no significant difference of expression of the NBC1 variants. So this was successful. And then I undertook some IF analysis, which can be seen here. On the top we have control cells. As you can see, it's barely visible because it doesn't have as much NBC1 doesn't express as much NBC1 as the other variants. Then we have wild type, which readily, you can see, expresses NBC1. 2 mutants that are included here, also expressing NBC1, and we can see with these results, the last 2 as well, that these mutations of the serine residues do not affect plasma membrane localization of NBC1. So, after this step, we decided we had to measure the activity of NBC1 variants relative to wild type. And how do we do this? I used an opera system, which has a chamber which allows you to adjust the temperature, humidity, and carbon dioxide levels. And by usage of 96 well plates, allows you to analyze many clones at one time. And it has different filters for emission. It has 3 cameras, which allows you to simultaneously measure emission at different wavelengths, several lasers for excitation. In this experiments I use SNARF, which is an intercellular pH-sensitive dye, which upon a 488 nanometer excitation, you can measure emission at 585 nanometers and 690, and the inverse relation will give you an accurate reading of what the internal pH of a cell is. The advantage of the system was I could analyze many clones at one time, but due to that, there's less time between measurements. So there's a trade-off between time resolution and high throughput. So the experimental procedure of the PHI measurements were first you load the cells with SNARF, and then you have to measure the activity of the acid extruder. You have to acidify the cell. This is done by the ammonium pre-pulse technique, which when you add ammonium, cells first alkalize, they load with acid, and upon ammonium removal, they're acidified. So their steady state pH is a lot lower than normal, and then they have to use acid extruding membrane transporters to return to normal levels of pH. So we decided to measure under the pH recovery rate, as it's called, under 4 different conditions. 20 micromolar IPA was always present to block actions of NHEs, and the 4 conditions was normal recovery where only IPA was in place, then addition of SRA59, which is a general MBC inhibitor. The third one was addition of DIDS, which is a non-specific bicarbonate transporter inhibitor. It's generally actually been described as non-inhibiting MBCN1 in most cell types. And the last one was sodium-free, where because both these transporters and other ones need extracellular sodium to work, there should be no recovery. I'm going to go through how the time course of how these experiments would look. On the top we have control cells, and on the bottom we have wild type cells. As you can see here, the first 4 points on each graph represent steady state pH, where we have around 7.5, which is a little bit elevated, but it's steady. The first arrow indicates addition of ammonium, through which cells first alkalize a little bit and then normalize over the minutes. And then the wash step, which removes the extracellular ammonium and acidifies the cell, as we can see here. This is the recovery phase. The black dots indicate normal recovery, and the red dots indicate sodium-free recovery. As we can see, initially in both sodium-free recoveries, cells are being significantly acidified to about 6, pH of scale about 6. For control cells, recovery can be seen here. It takes about maybe 5 minutes for it to recover. But for wild-type transfected cells, recovery is actually almost immediate. So within a minute, these cells have recovered. So the transfected MBC1 is working. So this was carried out for all the clones, in all the 4 different conditions I talked to you about. And here are some results. Here we have the 4 different conditions: normal recovery, SRA59, DIDS, and sodium-free recovery, where the recovery rates have been taken relative to wild type. Wild type is seen as the white bar, which is the control, always at 1. Interesting to note here in the start is control cells in the 3 first conditions have significantly reduced recovery rates, since they don't have as much MBC1. In sodium-free, this has normal recovery. There's no difference there. What I'd like to point out, significant in this first set of results, was that S1180 exhibited about 20% reduced recovery rate relative to wild type in normal conditions, and once SRA59 was added, this was extended to the S89A clone. I also took these experiments, these results, and analyzed them relative to the control condition per clone to assess the individual effect of each inhibitor. This can be seen here. The normal recovery buffer is where you have, this is the control condition, and you have the wild type and the 4 clones and the control cells here. What can be seen here is significant across the clones is that SRA59 induces about a 30% reduced recovery rate on each clone. DIDS induces about a 85% reduced recovery rate, and when there's no sodium, there is a lot lower recovery rate. So this is the initial results, and the conclusion for these results were that S89 and the S1180 clones had slower recovery rates compared to wild type, and S407A and S411A clones had normal recovery rates relative to wild type under the conditions. Another interesting conclusion I can draw is that rat MBC1D, which is heterologously expressed in these hex cells, was highly sensitive to DIDS. So we decided to go on further with this, and since these 2 clones were the interesting ones, we wanted to elucidate what's the mechanism behind this reduced recovery rate. So we stopped looking at those 2 mutants, and we only look at these 2. In the same study I talked to you about before,